So I'm going to tell you some recent findings from the lab, but in a context of what we have been doing over the last 10 years. So almost a little bit more than 10 years ago, we started deciphering how different extracellular cues could regulate pre-mRNA alternative splicing through the activation of different signaling cascades. And to do that, we started with a very well-known model for alternative splicing regulation, that is the FIBROMECTIN gene. I'm pretty sure you are all familiar with, but just briefly, this pre-mRNA or this gene contains 3 alternative regions that are shown here in red. And the combination of all these splicing events can give up to, can originate up to 12 different polypeptides in rodents and even more, up to 20 in humans. And in particular, we focus on the best characterized of these alternative splicing events, that is the inclusion or exclusion of one exon cassette called ED1 or EDA, which I'm sure you already heard a lot about it. And this event, the inclusion or exclusion of this exon cassette, can be easily analyzed by RT-PCR, as you can see here. So this is just a summary of part of our work. What we found is that conditioned media from mesenchymal cells, or that is soluble factors secreted by mesenchymal cells, or even certain purified growth factors that are from mesenchymal origin, stimulate a pathway involving RAS, PI3 kinase, and AKT that regulates the inclusion or exclusion of this exon cassette into the fibronectin mRNA. And the way it regulates it is stimulating the inclusion of this alternative exon. And here you see it on a gel after the PCR, and here the quantification. What we also found was that this signaling cascade was acting through 2 SR proteins, SF2 and 9G8, and that the phosphorylation status of these SR proteins changes upon the activation of this signaling pathway. And more important, that AKT is capable of phosphorylating these 2 SR proteins. And these results led us to propose that AKT can function as an SR protein kinase. And this was the work of Matthias Blaustein, a previous graduate student in the lab. So let's concentrate now in SR proteins, in particular one of these 2, that is SF2-ASF, currently known as SRSF1. And from work from many of you in the audience and many other labs, we know that this SR protein, in particular SRSF1, is much more than splicing regulator and have both nuclear and cytoplasmic regulatory activities. And here it is nicely shown in this cartoon. We know it regulates transcriptional elongation, genome stability, splicing, mRNA export, translational control. We have just heard a beautiful talk before the break, mRNA degradation, nonsense-mediated decay, and including pre-microRNA processing. So it seems like the list of roles for this SR protein, SRSF1, keeps growing. And a few years ago, my lab was able to add a new role to the list of this multitasking protein. And what we found was that SF2-ASF, or SRSF1, is able to regulate protein SUMOylation. And this was the work of Federico Pelic, that was a graduate student at that time in the lab, now is an assistant, research assistant in the lab. So what is protein SUMOylation? Protein SUMOylation is a post-translational modification that is the conjugation of SUMO, that is a small peptide from the ubiquitin family, to different target proteins. What are the consequences of SUMO conjugation? There are many. It's not a labeling for degradation as ubiquitin is, but what can do is to compete for acceptor lysins with other post-translational modifications. It can also interfere with protein-protein interaction, or it can enhance protein-protein interactions, or either intermolecular or intramolecular interactions. And this, what is shown here is the SUMO conjugation pathway, which is very similar to the ubiquitin pathway. First of all, SUMO needs to be processed in order to get into a mature form, and this is done by specific endopeptidases. Once SUMO is mature, it gets conjugated to the target protein by the action of two enzymes, an E1 activating enzyme and an E2 conjugating enzyme. Then there is another set of enzymes that are called E3 ligases that are not strictly required for this process to occur, but they help in the conjugation step. They facilitate the conjugation, and they provide substrate specificity. So they are very important in vivo, while they are not really necessary in an in vitro system. So this slide just summarized what we found. We found that SF2ASF is able to stimulate SUMO conjugation or SUMOylation in culture mammalian cells and also in vitro, and it displays certain characteristics of a SUMO E3 ligase. That is, it can stimulate the transfer from the E2 conjugating enzyme to the target, and it displays substrate specificity. That is, it works upon certain substrates but not on others. And in particular, we got interested by the fact that we found different RNA processing factors whose SUMOylation was regulated by SF2ASF. So knowing from Carmo's lab and from Manley lab that SUMO conjugation could regulate RNA editing and that also could regulate 3' end processing, we postulated that SF2ASF could be a link between SUMO conjugation and RNA processing machineries. And in particular, we wanted to look for a possible connection between splicing and SUMO conjugation. So to start with this search for this possible connection, we decided to start for our favorite signaling pathway that we have characterized that it regulates fibronectin pre-RNA alternative splicing. And knowing that AKT is an SR protein kinase that can phosphorylate SF2ASF, and having described that SF2ASF can regulate SUMO conjugation, we asked the simple question whether AKT could be a SUMO conjugation target. And so we first did a bioinformatics search, and what you see here, there are many putative SUMO conjugation sites within AKT. But it was important to see whether we could detect SUMO conjugation to AKT within living cells. So what we did was to transfect mammalian culture cells with these two constructs, one that expresses SUMO with a histidine tag, and the other one that expresses AKT with an HA tag. Then we purified SUMO conjugated proteins by nickel affinity chromatography, and we ran a Western blot. And with an antibody against the AKT tag, the HA tag, we could detect slower migrating bands that were retained by the nickel affinity column that corresponded to SUMO conjugation to AKT. So we then wanted to see which of the already known E3 ligases could regulate this conjugation. And what you see here in the third and in the fifth lane, that the overexpression of either PS1 or TOPORs, two well-characterized SUMO E3 ligases, can increase the conjugation of SUMO to AKT. And to others' surprise, also the overexpression of SF2ASF, which we have recently described, it functions in an E3 ligase-like function, could also stimulate AKT SUMOylation. So we then decided to map which were the bona fide SUMO conjugation sites within the AKT molecule, and we just did site-directed mutagenesis to each one of these sites. And I'm not going to show you all the data, but just summary of it. when we combine two mutations in these two putative sites, we get a strong decrease in SUMO conjugation levels to AKT. And this mutant, we call it 2KR because it has the replacement of two lysines by arginines. And with this 2KR AKT mutant, we kept doing many other experiments that I will show you in a minute. So a summary up to here, I have shown you about this signaling pathway that regulates fibronectin alternative splicing, leading to inclusion of the ED1 or EDA exon into fibronectin mature mRNA. And I told you that AKT can phosphorylate SF2. I also told you that AKT is a SUMO conjugation target. And I told you that SF2 can regulate SUMO conjugation to AKT. So our next question was whether we could get any consequences of AKT SUMOylation on alternative splicing regulation. And again, we got to the fibronectin gene and actually to the fibronectin splicing reporter mini gene that I'm sure you are all familiar with it. So what we did was to co-transfect culture cells with the splicing reporter mini gene and expression vectors for AKT, either wild-type AKT or the 2KR mutant that I just mentioned 2 slides ago that was deficient in SUMOylation. So what we found is shown here, something that we already knew, overexpression of AKT either under basal conditions or upon stimulation with a constitutively active RAS isoform increases ED1 inclusion levels. That's the wild-type AKT. However, if we do it with the mutant AKT, the 2KR that is SUMOylation deficient, we don't see that increase. That is, the activity of the capability of AKT to increase ED1 or to stimulate ED1 inclusion is lost when we mutate these two SUMOylation sites. We then did the opposite. We depleted endogenous AKT with an siRNA targeted to the 3' UTR. And as expected, we observed the same, a decrease in ED1 exon inclusion. And then we decided to rescue this inhibition by overexpressing AKT from vectors that were not sensitive to the siRNA inhibition. And what we observed is that we were able to rescue this SI-dependent inhibition when we overexpress the wild-type version of AKT. However, the mutant version, the AKT2KR that is not or is deficiently SUMOylated, was not able to rescue this effect. Then we got interested into a mutant AKT that was described in 2007 as a transforming mutation found, a somatic mutation found in many different human cancers. So this mutation is called E17K, and what it does is to generate a constitutively active AKT, that is an hyperactive version of AKT. It has a high capacity, a high transforming capacity in culture cells, and also a high tumorigenic capacity in mice. So we decided to check what was going on with this mutant AKT in terms of splicing regulation. And what we found is that in this hyperactive AKT was also hyperactive in our splicing assay, as you can see here compared to the wild-type AKT, strongly increases ED1 inclusion levels. We then checked the SUMOylation level of this mutant AKT, and we found that it was significantly increased. So we decided to introduce into this E17K mutant the two mutations in the SUMOylation sites that we have described earlier. So we converted into a triple mutant that has this mutation and the 2KR mutant that I have already explained. And to our surprise, introducing these two mutations in the SUMOylation sites completely abolish this hyperactivity of AKT. So this hyperactivity of the E70K mutant was absolutely dependent on its SUMOylation. So then we wanted to expand a little bit more and go outside fibronectin, and we check another alternative splicing event. And this is the BCLX splicing reporter that we got from the Chalfant lab. And as you may know, BCLX can give rise by alternative splicing to two isoforms, a long one that is a pro-survival isoform, and a short one that is a pro-apoptotic one. I need some water. So we did exactly the same. We transfected this mini gene together with the wild-type version of AKT, or the SUMOylation deficient version of AKT. And what we found again is that while wild-type AKT can increase the production of the BCLX long isoform, the mutant AKT, the SUMOylation deficient mutant, cannot do it. So AKT activation favors the production of fibronectin isoforms containing the ED1 alternative exon. These isoforms are characteristic of highly proliferative tissue. Also, AKT favors the production of pro-survival BCLX isoform, the long isoform, and it does so in both cases in a SUMOylation-dependent manner. So we know that AKT is more than a SR protein kinase. It regulates many other aspects within the cell, and this is confusingly shown here. AKT regulates many different cellular functions upon stimulation with many different extracellular signals, and it does so by regulating the phosphorylation status of more than 100 substrates. So we decided to see whether we can find any other consequences of AKT SUMOylation that were not strictly related to splicing, and we focus in survival and proliferation. And I'm just going to show you one result. This is the effect of AKT. AKT is known to favor the progression from G1 to S phase. And this is what you see here. If you overexpress AKT, wild-type AKT, the percentage of cells in S phase increases. However, if we overexpress the mutant AKT, that is the SUMOylation deficient mutant, that effect is no longer observed. That is, the known role of AKT as a stimulator of G1S transition is dependent on its SUMOylation. And this was the work of Guillermo Rizzo, now a postdoc in the lab, but this was his PhD thesis work, and this paper was published last month. And let me tell you that while we were writing this paper, another paper came out showing AKT SUMOylation and the regulation of cell proliferation and tumorigenesis. And although we got really upset when we saw the paper, then we got really happy because they basically show pretty similar results. So we are very confident that this is true. And so as a summary, I have shown you how the PI3 AKT pathway is involved in alternative splicing regulation, and how whether AKT function as an SR protein kinase. This is something that many other labs have already reported, and it has it has implicated AKT signaling in alternative splicing regulation, and this is just an example of many other papers that have been appearing in the last few years. So then we described that the SR protein SF2ASF has a novel role as a regulator of SUMO conjugation. And trying to combine these two pieces of data, we got to the result that AKT is modified by SUMO conjugation, and that this modification is involved in alternative splicing regulation and as well as in other AKT functions, adding an extra role of regulation for the activity of this key kinase both for normal cell growth and in the context of pathology. So for those of you who are interested in AKT, what we know about AKT post-translational modifications, since early 90s, we knew that it's modified by phosphorylation. Around 2009, it has been reported that it's also ubiquitinated, and there are two types of ubiquitinations on AKT. Some of them are activating ubiquitinations, others are just labeling AKT for degradation. From 2011, we know that AKT is a substrate for acetylation, and actually acetylation inhibits AKT activity. It requires to be deacetylated in order to be activated. And since this year, we know that AKT is also SUMO-related, and as I have just shown you, SUMO conjugation is involved in the regulation of AKT activity. And to end, I want to thank the people involved. This is the current lab, Pablo, Berta, and Guille. Federico Pelic is currently in Dundee for a couple of years. Past members of the lab, collaborators abroad, collaborators in Buenos Aires. This is our funding, was our funding, now the list is shorter than this. And I really want to thank two people in the lab that are absolutely required for daily life. That is Valeria Buggiano, our technician, and Alberto Kornblit, our inspiration model. And I want to thank you, everyone.